Okay, I would like to welcome Dr. Victor Gomez-Leon. He's the assistant professor in dairy and an extension specialist. Dr. Leon is a recipient of a 2020 Global Food Systems Initiative Sweet Seed Grant. And the title of his presentation is Effective Diet Supplementation with Omega-3 on Reproduction and Milk Production of Lactating Holstein Dairy Cows. Dr. Leon?
Okay, thanks for the presentation. So it's a great honor for me to be one of the recipients of this funding, and it was a great honor and a great opportunity to conduct this research, especially because it was my first year at K-State. So exciting times. So that's the title of our presentation. Again, I am the PI of the project in a collaboration with Dr. James Rullard, and the master student leading the study is Santiago Paez, who is started last year in our program at K-State. So we wanted to conduct this study because there are some studies in the literature that have shown that feeding omega-3 to dairy cattle can improve pregnancies per insemination. And so pregnancies per insemination is not just about reproduction. It also helps the health of the cow, because when they get pregnant at the best time, they will have less health problems postpartum. They will produce more milk, etc. And so that omega-3 has been related with increasing pregnancies per insemination, which has been associated with the corpus luteum, with less regression of the corpus luteum. And so the corpus luteum is the structure that maintains the pregnancy. But although some studies has been conducted and shown that there is an effect on the embryo in vitro, there has not been any studies say for the in vivo embryo development or the pregnancy development. And so that was one of the objectives of our study. And also, it is very important for us that work with livestock, of course, producing milk, feeding the world and everything. But cattle is really a great model for studying human reproduction. So for the last couple of years, we've been studying pregnancy losses, especially from that first to the second semester of the month of the pregnancy. And so this is a great model to address on effects of omega-3 on that embryo or conceptus development. So again, our objective was to assess the effect of omega-3 on milk production, but also on pregnancy-related parameters in lactating dairy cows. So our hypotheses were that feeding or increasing omega-3 fatty acid on lactating cows will increase its concentration in milk, enhance the oocyte quality, so the egg quality that will develop the pregnancy later on, and also increase or enhance parameters associated with the pregnancy per se. And so let me explain what we did on the study. We basically divided the study in two phases. Phase 1, you can see here on top left side of the screen, phase 2 on the bottom. So on day 15 postpartum, we randomized cows to a control group or an omega-3 group. And what we have are two different diets. And you can see based on table 1 here, the greater plus is the group that is rich in omega-3. The other commercial fatty acid is rich in omega-6. So I have to mention that feeding fatty acid is a common practice for the dairy industry. But what we are doing here, what we are doing in this study is changing the profile of the fatty acid. So you can see linoleic acid, 50% of the content of this grade of plus, that's omega-3, versus 0.2% of omega-3 in this other commercial fatty acid, and palmitic acid, 50% on these other commercial fatty acids, which will be omega-6 content. So we are switching omega-6 by omega-3 to see the effect of that into the milk production and reproduction. So we start that at day 15 postpartum. Those cows were start eating those diets, and on day 50 we start we collected oocytes. So we collected eggs or from the ovary of these cows to see the quality of those oocytes. On day 75 or 110 days postpartum, depending if they got pregnant to the first or the second insemination, we bred those cows, all of them, and starting 11 days after the insemination, every week until day 60, we evaluated the corpus luteum volume, the embryo, and the vesicle volume to see if we were going to improve the quality of those pregnancy-related parameters. So we have in the study primiparous and multiparous cows. So first and second or older lactation cows. We divide them into 25 cows in the control and 25 in the omega-3 group. This is a pen study, meaning that the experimental unit is not the cow, is the pen. So we have a group of cows in 3 different pens per diet. We feed them, they have unlimited access to water. They were fed only once per day. Again, that's also common for the dairy industry. And they were milked 3 times per day, which is common for the Kansas State University Dairy Teaching and Research Center, which is the place where we conducted our research. So let me continue to show some of our results. So we wanted to address different questions. And of course, the first question is, did the treatment really work? Did we, by feeding more omega-3 in the diet, did that increase or maintain omega-3 in the blood sample in the system of our cows? So what you can see in this graphic here is the ratio of omega-6 to omega-3 in numbers here in the y-axis, and days in milk or days postpartum in the x-axis. You can see in the green dots the control group, the average for the control group, and in the purple squares, the average for the omega-3 group. So you can see in day 15, they start at the same level, but as time passes, the control group, if eating more omega-6, they increase the ratio of omega-6 to omega-3. And the omega-3, it decreases a little bit and then just maintains. So if we have a higher ratio of omega-6 to omega-3, that means that we have lower omega-3 in the diet. So we could say, yes, we maintain a lower omega-6 to omega-3 ratio from days 50 to 140 days in milk. So a very representative period there for a cow that is producing milk for 305 days. Of course, common sense question was, well, that was in plasma, that was in blood. Did that translate also to the milk? So did the diet affect the milk fat profile? And so you can see here a more direct comparison. This is actually omega-3 percentage in milk. And we have the groups by diet and days in milk. So 15 days in milk or 15 days postpartum and 50 days postpartum. And you can see again that at 15 days, they start both at the same amount of omega-3 in milk. But then by day 50, the control just decreased the amount of omega-3 in milk, whereas the cows receiving omega-3, they just maintain that level. And so, yes, we also maintain greater omega-3 in milk at day 50, which was one of the objectives and hypotheses of our study. So we're very pleased with that. This is also a very similar comparison, but now it's actually the same comparison, but now what we are seeing here is actually the amount of omega-3 in milligrams that will be contained in a 8-ounce glass of milk. So you can see that by day 15, they are again the same, but by day 50, they are different. So if you drink milk from one of these cows on day 50, you will get about 50 milligrams of omega-3 in that 8 ounce of glass of milk, whereas here you will get about 30%, which is a considerable difference for those two diets or those two type of milk. It's coming from the same cow. They are eating the same amount of fat. It's just we are just manipulating the amount, the profile of the fatty acid. And so, of course, we wanted to know if something else also changed in the milk content of this. So when we are working with dairy cows, we measure different things like total fat, the amount of lactose, urea in the milk, the amount of protein, somatic cells, other type of solids. And so if you can carefully go through all these probabilities that are down here, you can see that there is really no probability that is significant that is related to the diet content. The probabilities that were significant were related only to days in milk or days postpartum. So meaning that our diets didn't change any of these other milk components. And that's good because that was one of our hypotheses as well, that we were not gonna change the component of the milk, but we were gonna change how much or the fatty acid profile of the milk. So that's good. But then we also have to ask the question, well, did we change the amount of milk that the cows were producing? And so this is a more busy graphic, but we have here milk production in pounds, and we have days in milk or day postpartum in the x-axis. You can see in the graphic, these two lines here, the squares will be primiparous cows. Again, primiparous control in green, primiparous omega-3 in purple. And so the first thing you can notice is primiparous, they produce less milk than multiparous cow, correct? And you can see here the parity effect being significant. But there was not there was a tendency for a diet by parity by days in milk interaction. And that tendency was coming from these cows, the omega-3 multiparous cows, producing more milk over time compared to these control multiparous cows, okay? And so multiparous cows eating omega-3 had greater milk production or had a tendency to produce more milk production, which was not intended, but that's how the research result came. So then the question was, well, maybe could it be possible that these cows producing more milk are really producing more because they are eating more? And so we also analyzed feed intake in this study. So every week we clean the bones of the animals, we put a specific amount of feed, and the next day we will go and take the leftovers and weigh that so we could know how much they ate. And what we see here is the feed intake per pen. And remember that the experimental unit is the pen and not the cow. So about 12 cows per pen here because we have other cows there as well. And so we have control primiparous, control multiparous, omega-3 primiparous, or omega-3 multiparous. And there is no difference in how much those cows were eating. So they were all eating the same. And so with the same feed intake, these omega-3 multiparous were making more milk than the counterparts on the control multiparous cows. So that was interesting to see as well. So we kind of went through the milk data, and it was interesting. We were able to support our hypothesis. And then we moved to the reproductive metrics. So in this table, what we have is the data related to cyclicity postpartum. So in other words, after the postpartum, how soon those cows start to cycle, start to ovulate, and have cycles that could be fertile. And so we have here, again, divided by control or omega-3s, primiparous and multiparous, and we have the total. We analyze the prevalences of cysts. Cysts are structures in the ovary that do eggs that do not ovulate. And so no difference there. We also analyze the largest follicle size, so the largest egg size, because it's related to an earliest ovulation. So the greater, the better. We also don't want a huge size. So this is in millimeters, so about 18 to 20 millimeter is a good structure, but there is no difference on those largest or second largest follicle size. But in days in milk, we were able to find the largest follicle at an earlier time in the multiparous omega-3 cows. In the primiparous omega-3 cows compared to the primiparous control cows. So it seems that we help a little bit on this omega-3, but not much difference really. It's just a tendency, so we don't want to make a big deal from this. Now we analyze the quality of the oocytes. Remember I told you we went and aspirate the eggs from the ovary. And again, this is another very populated table, but it's divided by control and omega-3s and primiparous or multiparous. For dairy cows, we are always analyzing data differently for primiparous and multiparous because it's kind of a total different animal. The physiology is different. And so there were no difference in the number of aspirated follicles per cow. So the number of eggs per cow was not different. The average or the quality of those eggs, when we saw grade 1, grade 2, grade 3, and grade 1 is the best, it goes, then grade 3 will be the worst or degenerated. There was also no differences when we analyzed those categories separately. You can see the p-values here on the far right, the effect of diet parity or diet by parity. But when we analyze the grade 1 and grade 2 together, there was a tendency for the animals on the omega-3 group, multiparous cows specifically, to have a better quality, to have grade 1, more grade 1s and grade 2. And so we combine grade 1 and grade 2 because those are the two categories that we take for the culture, for the in vitro culture. The grade 3 and the degenerated, we usually don't take. So again, the omega-3 help those multiparous cows to have a better quality of oocytes, but really no much differences in the controls versus the primiparous omega-3. In reproduction, we also analyze the CL size, so the corpus luteum, which is the structure that produces progesterone to maintain the pregnancy. So we analyze, this is the CL. That's the way you will see it in an ultrasound. The color is to analyze the vascular perfusion, or as we call it, the blood flow, how much blood is coming to that CL or going out of that CL. And so in this analysis, what we used was all the cows that were bred, so all the animals, regardless if they were pregnant or not. And this is the data related to day 11 and 18. So for dairy cattle, we do pregnancy diagnosis with day 32. So this is before pregnancy diagnosis, before knowing who is pregnant. And unfortunately, there were no difference related to the diet. You can see here the diet, there's no effect. And all these interactions related to the diet are also not significant. So there was not an effect of the diet on the CL volume or the blood flow, the CL blood flow. And those two things are related to better quality of the pregnancy. So no difference there before knowing the pregnancy diagnosis. Once we just analyze the animals that were pregnant, we can see this graphic here. So this is again CL volume in millimeters and days post-insemination. And we have now control primiparous, the green squares, control multiparous, the circle green, and omega-3 primiparous, the squares that are purple. And so you can see how it's very similar across the day for those three groups. Consider that we have 6, 9, or 8 cows for each of those groups because these are only the pregnant animals. we remove some animals with twins and things like that that may affect the CL or may affect the physiology of the animal. So again, no effect of group or diet. There is an effect of time and an effect of diet and daisy milk. Probably because of this, we did a separate analysis, and this the corpus luteum volume on the omega-3 primiparus was bigger than the corpus luteum volume on the control primiparus, but it was similar to the control multiparus. So on day 11, it seems that, again, we help a little bit the primiparus cows, the corpus luteum there. And so that's maybe related as well with improving the pregnancy. But again, it was just a tendency. Now, when we see the vascularity or the blood flow of the CL only on those pregnant animals, what we can see is that it's very consistent across the time, but there are some days in which the blood flow or the vascularity of the CL drops in some of the control primiparous cows. And the omega-3 seem to help to maintain that blood flow very constant during those times, which are 25 and 39. And interestingly, 25 is a day where we'll have cows returning back into estrus. So maybe there is some physiologic explanation for that. And then 39 is also another physiologic time where we see some pregnancy losses occurring. Not, again, this is just data for maintained pregnancies, but physiologically, this is a time where we see some pregnancy losses start to occur in other trials. So it seems that we help to maintain the blood flow, the vascularity on some of those CLs in primiparous cows receiving omega-3. Now, the other thing that we measured was the embryo. You can see here in the image on the right, this is a pregnancy in dairy cattle. So in yellow, you can see the embryo per se. And in blue, you can see the vesicle that contains the embryo. So we measured those two parameters in all the animals, in those 6 primiparous, 9 multiparous control, and 8 primiparous omega-3. But there was no difference of group or group by days post-insemination, not for the embryo size and not for the vesicle size. So what I'm showing you here is just the average for all those cows, for the 23 cows that were pregnant. And so you can see how the vesicle size, it's very similar to the embryo here, as you can see in the image here on the right. And then they both start increasing, but the vesicle just increased faster. But again, we couldn't find an effect of the diet on those two parameters. So not affected by diet or parenting. As final considerations, we have that our results demonstrate that we have some improvement in the quality of the oocytes at 50-day postpartum when we fed cows with omega-3. Now, we don't know if that it's maintained until 140 days. We based on the blood samples, it is maintained until that time. But we also can speculate a little bit that this effect at 50 days in milk may help producers that start breeding their cows postpartum very early. So at 40 or 50-day postpartum, when we start breeding animals at that time, one of the problems we have is the oocyte quality is poor because the cow is producing a lot of milk. And so putting a lot of energy towards the milk and not towards reproduction. So it seems that feeding with omega-3 may help a little bit on that issue, but we need more research to really support or not that observation. And so a direct effect of the omega-3 on the embryo and the vesicle was not supported based on our results. However, we seem to have affected the CL, the corpus luteum that maintains the pregnancy at some specific days based on the blood flow on days 25 and 39. And I feel that perhaps we need a little bit more of data to rule out this lack of an effect on the embryo and vesicle development. And finally, we also support the hypothesis that we will increase the omega-3 in blood and milk of these cows. So while we are helping this cow to achieve more omega-3, maybe even have a healthier system for their own, we may be able to produce a product that is more amicable for consumers and that will attract more people, because it's omega-3 that is coming already in the milk and not added afterwards. So as an acknowledgement, I have to thank, of course, the Global Food Systems Seed Grant Program for their funding and support. It is a great initiative. The Naturally Better Omega-3 Technologies is a company from Manhattan that matched the funding for this study, and all the collaborators within and outside K-State. So with that, I'll be happy to, if you have any questions, please send me an email. I'll be very happy to entertain and reply to any of those.
Thank you so much, Dr. Gomez-Leon. And as he stated, if any of you have questions or would like to discuss this technology further, please feel free to reach out and have that talk with him. So thank you.